Processed meats such as bacon, ham, sausages, and deli cuts have come under intense scrutiny for their link to cancer, particularly colorectal cancer. Central to this concern is the use of nitrites, which are added to these meats to preserve color, prevent bacterial growth, and enhance flavor. While nitrites themselves are not inherently carcinogenic, the conditions in the human body, especially within the digestive tract, can transform them into a far more dangerous class of compounds, nitrosamines. This conversion is the key biochemical step that helps explain why even modest consumption of processed meats has been repeatedly linked to cancer risk. When consumed, nitrites can react with amines and amides, naturally occurring components of proteins, in the acidic environment of the stomach. This chemical reaction leads to the formation of N-nitroso compounds, including nitrosamines, many of which are potent carcinogens. These substances can damage the DNA in colon cells by forming adducts, structural changes to DNA that disrupt replication and repair processes. Over time, this DNA damage can accumulate, increasing the likelihood of mutations that drive the development of cancerous cells in the colorectal lining. The risk is not purely theoretical. Experimental studies have demonstrated that nitrosamines can induce tumors in various animal models, and human epidemiological data strongly supports the association between processed meat intake and colorectal cancer. The International Agency for Research on Cancer, IARC, has classified processed meat as a Group 1 carcinogen, meaning there is sufficient evidence to establish a causal link with cancer in humans. Importantly, the risk appears to be dose-dependent. The more processed meat consumed, the higher the risk, though even small amounts consumed regularly over years can elevate lifetime cancer risk significantly. Additional factors may exacerbate this pathway. Cooking processed meats at high temperatures, such as frying or grilling, can further increase nitrosamine formation. Meanwhile, processed meats often lack the protective compounds found in plant-based foods, such as fiber and antioxidants, which can help reduce the impact of carcinogens and support DNA repair. Diets low in fiber may also slow transit time in the colon, giving nitrosamines more opportunity to interact with and damage intestinal cells. Despite the biochemical risk being well established, nitrites continue to be used in food manufacturing because they serve important practical functions. Some newer products now use natural nitrite sources like celery powder, but chemically, these nitrates and nitrites behave in similar ways once metabolized. So labeling a product uncured or no nitrites added does not necessarily mean it is free from nitrosamine-related risk. Understanding the nitrite to nitrosamine pathway shifts the conversation from vague warnings about processed meat to a clearer picture of why this category of food is risky. It's not simply that these meats are fatty or salty. It's that their preservation process introduces chemical precursors that, under common biological conditions, transform into known DNA-damaging agents. This mechanism is biologically plausible, reproducible in lab settings, and strongly supported by population-level data. Reducing intake of processed meats is therefore not just a matter of general dietary improvement. It's a specific strategy to reduce exposure to a well-characterized carcinogenic pathway. While occasional consumption may not pose dramatic short-term danger, consistent, long-term exposure quietly increases risk in a way that's easy to overlook. Knowing how the process works empowers consumers to make choices not just based on taste or convenience, but with a deeper understanding of what those choices mean for long-term cellular health.